So let's say we don't want to be a single-celled organism. We kind of want to be a trillion-celled organism that we are as humans. It just makes life a little bit easier to be able to think and process and not just bump along and hope that you got the right resources. So what you got to do is you got to replicate your DNA. You've got to have more than one cell type. You've got to have more than one cell. So what you go through is mitosis. Mitosis is the process of getting the cell ready. How it does that. You'll notice that mitosis isn't really much more than maybe fourth, maybe an eighth of the time that a cell will dedicate its life to. It could even be less than that. If it decides it doesn't really want to replicate, aka thanks brain cells that don't want to keep on replicating, which I guess is a good thing because I wouldn't really want my brain to explode. Anyways, sorry. G0 is the resting phase. It's not dividing. It's happy where it is. We don't need any more. If it's decided, you know what, we need more cells, you're going to go through several phases of interphase. Interphase is the before phase of all of mitosis. G1 phase, the G1 phase is the description of you're making more organelles, you're making other bits and pieces that both new cells are going to really need. S phase is the formation or the creation of DNA. This is the phase that you're going to actually duplicate DNA because you really need, both cells really need a copy of that DNA. You don't want one missing a piece or not, because if it does, then it's probably just going to either break down or in the rare case become cancerous. G2 phase is when you're getting more enzymes and proteins and those type of things made. Once all that is done and everything, the stage is set, then you can go through the things that you actually see like pictures of when you think of, or things that you think of when you're going through mitosis. So interphase, again, G1 phase, you're making organelles. S phase, you're duplicating DNA. G2, you're making the proteins. Note some structures of it. Please, if you see a photo like this or this, try and memorize it. You'll notice that the inside, this nucleus is still formed. It still has an organelle. You don't see any DNA condensed. Maybe you'll see the nucleolus maybe in the middle. Who knows? All of that's intact. It looks like a nice, happy, normal cell. Mitosis, on the other hand, has 4 other stages that play a role of what's going to happen. Or you can see it and you go, yeah, that's definitely this phase. That's definitely that phase. That's what's going on here. Prophase is when, you remember how I said that certain DNA doesn't really like hanging out condensed? Well, this is the exception where it wants to be condensed. It wants to condense down. It doesn't want to be super fragile, and it doesn't want to show off its genes quite yet. They're very nice blue genes, but they don't want to show it off. Anyways, they've condensed down. You'll also notice that the nuclear envelope has been decimated. It no longer exists. So if you see condensed chromatids that look like little hairballs stuck in the, just smack dab in the middle, no nuclear envelope, then you could say, hey, that's prophase. The next one is called metaphase. Metaphase is, well, meta. Meta means smack dab in the middle. So a metaphase plate. So your chromatids, your chromosomes, have lined up smack dab in the middle. They are happy where they are. That is metaphase. If you see these chromosomes all lined up so pretty, awesome. You'll also notice that the centromeres are hanging out on polar opposite sides. If you remember back to our discussion on microtubules, remember microtubules are the transport network, and you're going to see those networks finding their target on these chromosomes and linking onto them. Once they're all linked up and they're ready to roll and they get the signal, boom, anaphase happens. Anaphase starts ripping apart the chromosomes. So you're left with a pair of chromatids on either side. If you were to, what that looks like, it looks like, well, metaphase, but somebody's being a jerk and put some soap in the middle and it's moving to either side. Telophase, you'll actually start seeing the nuclear envelope start to form, maybe. You also see that the end of anaphase and the formation of a new cellular envelope or cell membrane. And cytokinesis, which isn't really considered a part of mitosis for a reason or another, is just the complete separation. And they become new individuals. They become sister cells. They're called sister cells because, well, they're identical. So, how that works, I don't know. Somebody has a backstory. Now, how I remembered that order, interphase, prophase, metaphase, anaphase, telophase, is, well, it's kind of dorky. But it helped me remember it, and hopefully it'll help you remember. It's kind of like you have a sibling, a really, really young sibling that doesn't know personal boundaries and all that. And they're so very proud of an accomplishment. They're at that age where they're potty training now. And they walk, and your name's Matt, by the way. Congratulations. Hello, Matt. So your sibling walks up to you, Matt, and says, "Hey, Matt, I pee, Matt. I pee, Matt. I pee, Matt." Interphase, prophase, metaphase, anaphase, telophase. Thank you, little brother or sister. I love hearing about how you've learned how to pee. Oh, Matt, I didn't pee in the toilet. What? So, control of cell density, what determines what happens and why it's around? A lot of the time when you go through mitosis, it's for tissues that don't really survive very well. So the inside of your mouth, a lot of the times when you're eating stuff, a lot of the stratified, those cells in there get rubbed off. Your skin dies off because it gets too far away from the food supply. Other places such as that aren't so abrasive won't go through that mitosis so much. The male gametes, aka sperm cells, will often and frequently reduplicate and will continue to do their thing. But I digress. Now, I did mention the male gamete because there's actually an important thing and actually the female gamete does this too. There's something called mitosis, and then there's something called meiosis or meiosis. Depends on how you like to say it. I like meiosis or meiosis. It's my friend. It's pretty much just a double-decker double-decker mitosis. You get one phase, IPMAT, interphase, prophase, metaphase, anaphase, telophase, but you get a second set. Those same cells that just divided are going to go ahead and divide again. So you get an anaphase, metaphase, prophase, metaphase, anaphase, telophase, but twice. Something to note really quick about some other differences that you'll notice up here in prophase, there's something called crossing over. What happens is these guys come along, they hold hands, but all of a sudden, oh no, Betty, Bertha, Ross, we're never going to see each other again. How will I ever remember you? Well, my dear, take my arm. Oh, Rob, thank you so much. And that's a really weird way. Usually it's just like in band camp, you trade maybe a trinket or two. But they're trading their arms. They're trading their legs. So maybe they're just trading shoes. Let's look at it in a less morbid way. So they trade off a piece of their genetic information. They're no longer identical chromatids anymore. And so crossing over occurs in prophase. Prophase 1 of meiosis. And that also explains a little bit as to why every sibling is a little bit different from one another other than being male or female. While a female sibling will be a little bit different from another female sibling is in part this crossing over. So at the very end, you're going to have quite a few differences between these two. First off, you're going to notice if you use this as your comparison, you're going to notice that there's quite a bit less chromosomes here. This is a diploid number of chromosomes, meaning that they still have a pair of those chromosomes going on. The chromatids going on. They still will be able to form an X. Well, these guys over here, they've not only split off an X, but they've gone ahead and split that X in half. And now it's just a line of stuff. And they won't be able to replicate unless another similar cell, like from a male to female or female to male, comes together. They're not going to go ahead and go through mitosis again or meiosis again. They'll probably die off. If I were you, here is a little makeup slide show that I came up with for you. This is the answer sheet to the mitosis for meiosis. These are the things that you do need to know about it. You'll notice that mitosis goes only in somatic cells, meaning body cells. So everything except for the gametes, aka the sex cells, the eggs and the sperm, will go through mitosis. Mitosis, you'll see that how many times does it go through PMAT, prophase, metaphase, anaphase, telophase. There's one phase here, two phases here. Does mitosis have a crossing over event that says they share trinkets with one another? No, they're cold stone. They don't care about each other. It's like, goodbye, loser. While meiosis is like, oh, we were best friends. Here's a trinket. Are the cells identical at the end? Yes. In mitosis, yes. In meiosis, no, not so much. Mitosis, number of cells at the end, well, 1 to 2. I don't know why I put 1 to 2. 1 possibly because one might die or get sacrificed. While meiosis, you're going to have 4. Is it diploid or haploid? Diploid for mitosis, meaning that it has a chromosome intact, while a haploid, meaning that you've cut that X in half and you only have one part of the tail. Chromatids, so again, because you have a pair, you have 46, but chromatids only 23. You only have one copy of each. That could be a little bit dangerous. But hey, who am I to judge? Let's go ahead and finish off this. So because of that display of this information and how DNA is expressed, you can form a whole bunch of different cells, each of which doing something. And the exact same DNA information is used in different ways and different strategies to be able to do what they can do. Nerve cells, even though they have the exact same genetic information as a red blood cell, are still nerve cells because they only express specific genes. Now, as we age, our ability to maintain mitosis and make certain the functionality of it all could start going to pot. It could just not do it very well at all, not do it, or maybe it's the checks and balances behind it could go wrong. So in that case, when cells start dividing uncontrollably, that's called a cancer. There's different types of omas, I'm going to call them, or emias. Pretty much, if you see an oma at the end of it, it's cancerous. So melanoma, sarcoma, osteogenic sarcoma, lymphoma. Pretty much the very first part of the cancer tells you where it came from. Melanoma, this is a melanocyte. It's a cell in the skin that produces pigment. Sarcoma, well, it's a cell that has a lot to do with, I'm having a brain fart on this one. Right, sarcoma is pretty much any of the connective tissues, or like something's going wrong with your I can't believe I'm doing this during a recording. The softer tissues, the connective tissues like ligaments, tendons, inside your joints, things like that. Osteogenic sarcoma, osteo refers to the cells in a bone. So osteogenic means the production of these cells. You got leukemia, which is a blood cancer, and lymphoma, which means that there is something wrong with your lymph nodes. The growth and spread of these cancers just depends, really, with really how quickly they are out of control. Some cancers can be really scary and take out a person in a matter of a month if it's not taken care of, while other cancers could have been going on for years undetected, going extremely slow. Each of which, it depends on what causes it. Usually it's some type of malware, I'm thinking computers. It's some type of, maybe oxygen did something with it. Maybe it's destroyed DNA and the DNA has tried to do its very best to do its thing. Usually the cell will try and pop if it's something, but detect something like that, but every so often it doesn't do that. It's kind of hard to commit suicide. Treatments, well, not a fun way to go. Most of the time, historically, how we've been doing that is doing a biopsy, pulling it out. If it's spread or if it's possibly spread, we're going to do some chemotherapy that targets growing cells. So like cells such as, that's why your hair often falls out, or it feels like your skin is on fire, because your skin is always growing. And since this chemotherapy isn't really targeting a specific thing, it's only targeting cells that are growing, then those cells can die. Those cells that are growing can die or get damaged or get hurt. That's why it's so painful. That's why it's so, sometimes, a lot of the times, people lose their hair, and it's very possible that it doesn't grow back. With that, that is the end of our discussion. Thank you very much. If you want to, you might want to go grab those makeup slides and test your might to see if you understand it. And I will see you next time.